All right, everybody. We're going to start with a second seminar, my second guest for the day. Once again, my name is Professor Veitrai, and I'm delighted to host Dr. Heidi Hirsh, who's visiting us from Mountain View, where she's currently working for CIMAS and NOAA remotely, and has finished her PhD in 2020 from Stanford, and she is a marine biogeochemist. Her PhD was on the spatio-temporal variability of biogeochemistry in kelp forests and in Palau?
Seagrass.
With seagrasses, yes. And we're just, delighted to host her here. She's working on her postdoc through end of 2022, and we'll be sharing some of her research that she's conducted so far. So take it away, Heidi.
Cool. Thank you so much, Vade. Like Vade said, I'm Heidi Hirsh. I will be presenting some of my research, both from my PhD as well as my postdoctoral work so far, on understanding local drivers of coral reef biogeochemistry. I'm going to share some data from a Palauan seagrass and coral environment that I researched during my doctorate, and then show some preliminary analysis and modeling from the NOAA National Coral Reef Monitoring Program data that I've been working on in my postdoc with Dr. Ian Enochs, hi Ian, and Dr. Tom Oliver. So if we move forward, I'm really interested in understanding what the variability is of biogeochemistry on local scales, with the utility of applying that knowledge to better understanding how these sites may respond to environmental change and stressors over time. And the first step in understanding that variability is, of course, to identify the stress or the drivers that change biogeochemistry over time. So those are things like our benthic composition, things like the physical environment, our tides, velocity. And then once we know what the drivers are, we can start to identify their contributions to a site's resilience or vulnerability, especially in the face of change. After we have a better understanding of these contributions on a site-by-site basis, we may be able to inform local management and solutions on local scales to start addressing these bigger global problems at a site-specific level. The problem that I think a lot about is ocean acidification. So I think everybody's pretty familiar with OA, but I'm going to provide a brief review. We know anthropogenic CO2 emissions are increasing our atmospheric CO2. And as you can see in this plot, this is data from Hawaii showing those atmospheric CO2 concentrations in red. And then we also see this concurrent increase in seawater carbon dioxide as those atmospheric concentrations increase. And this is because we have this equilibria at the ocean surface, driving those increases in ocean CO2, which in turn drive a system of chemical equilibria, which ultimately leaves us with more free hydrogen ions in the ocean. This increase in hydrogen ions drives lower pH or more acidic water. So we have this increase in acidity, which is also accompanied by a decrease in our carbonate ion concentration. And this is what's physiologically relevant for calcifying communities that need CO3 2- or carbonate to build their calcium carbonate shells or skeletal materials. Another primer before we move on into some data. I think most people are familiar with the carbonate chemistry system, but I just want to provide some reminders of some of the metrics that I'll be talking about today and show how some of these measurements that we think about when we're thinking about the carbonate system relate. So again, we have this system of chemical equilibria where dissolved CO2 is equilibrating at the ocean surface and then driving the system of reactions that give us carbonic acid, bicarbonate, carbonate in different ratios. And we track the available carbon through a measurement called dissolved inorganic carbon, or DIC. We also track the charge balance of the seawater, or alkalinity. And two of the components of DIC, that bicarbonate ion and carbonate, are included in alkalinity. They would be our carbonate alkalinity. You see that carbonate has that double negative charge, so it counts twice towards alkalinity. And then finally, we track the availability of our carbonate ion through aragonite saturation state, which is the saturation of carbonate in the ocean, or sometimes you'll hear it referred to as omega. And again, this is really relevant for these biological processes, especially on coral reefs. So I've mentioned calcification a couple times, or calcification and the importance of the carbonate ion In the calcification and dissolution reactions, calcification removes that carbonate and builds calcium carbonate skeletal material or shells, and then the reverse reaction would add carbonate to the system. The other metabolic process that we think about a lot is photosynthesis and respiration, which of course in photosynthesis, we're taking up carbon dioxide and producing oxygen, and then the reverse is true in respiration. We can look at how these metabolic processes affect biogeochemistry through a property plot of DIC and alkalinity. So now those three metrics that we talked about on the previous slide, we can see how they all work together in this figure on the right, where we have our salinity normalized DIC on the x-axis, salinity normalized alkalinity on the y-axis, and then our backdrop is that aragonite saturation state, where the higher saturation is in blue, lower saturation, which is our more acidic environment, is in red. And you can see these metabolic processes plot on top, where calcification moving down, dissolution moving up, affects alkalinity and DIC at a 2-to-1 ratio. And this is because, again, that double negative charge on carbonate reduces alkalinity during calcification. You're also removing a component of DIC, so you get that 2-to-1 ratio. And then photosynthesis and respiration, of course, affect our DIC because we're changing that concentration of carbon dioxide, but they're not affecting our charge balance. So we only expect to see data kind of moving around along this horizontal axis and not affected by alkalinity, or sorry, by calcification or dissolution. As Vade mentioned, my PhD focused on the variability in space and time in local biogeochemistry in kelp forests in Central California, and then in the seagrass environment, which I'll be sharing with you today. And I'm really, or I was really, and I still am, interested in this capacity for these environments to provide some local protection or mitigation against ocean acidification stress. And this is something that's been embraced in the scientific community as well as among policymakers and managers. This is a report published in 2018 by the California Ocean Protection Council and California Ocean Science Trust, summarizing some of the knowledge at the time about this role for seagrass and kelp communities. This report included this figure showing potential carbon pathways in these environments. And I'm not going to go into blue carbon and all the different carbon pathways, but I put this up to show the similarities in these systems of primary producers. And I want to highlight the role of photosynthesis and respiration as it impacts carbon. So again, we showed this reaction before, but in the forward reaction of photosynthesis, we see this drawdown in that carbon dioxide, which buffers pH. So we expect to see that increased local pH signal when daytime photosynthesis is happening. At night, however, we have the reverse reaction of respiration, which is actually adding carbon dioxide to our system and can further acidify that environment, making it potentially more stressful. In addition to the metabolic modification provided by photosynthesis and respiration or calcification dissolution, we expect the physics to also drive the signal that we're able to observe in terms of biogeochemical variability. So if we have a high residence time in a community, we expect that signal to manifest larger as there's more time for the community to affect that water mass. Whereas if the current is really ripping, we expect that signal to be dampened since there's not a lot of time for that water to be affected. Similarly, tidal volume will be a driver. So we expect at high tidal volume, or high tide, that signal is going to be diluted over a larger volume of water, whereas at lower volumes or lower tides, we expect to see a larger signal per unit of water volume. So as we're looking at these coastal sites, we expect that variability in terms of our chemistry is going to be driven by a combination of those benthic processes as well as our hydrodynamics. And in terms of hydrodynamics, we may expect the biggest metabolic signals at times of high residence time and lower volume. So I'm going to show you some data from Palau, where again, I did part of my PhD work. In the following slides, I'll show you more pictures, but this is the seagrass and coral environment I'll be talking about. The objective of this work was to capture that variability in this specific site, both looking at variability over time, over 16 days, and then over a transect in space. to better understand the role that seagrasses were playing as photosynthesizers in potentially mitigating that acidification stress or boosting our pH. This work took place in the Republic of Palau, located in the southwestern Pacific in this red box. My specific study site was in Koror State at the entrance to Nurmid or Nico Bay here. So if we zoom in on that, I designed a transect at the mouth of that bay along the axis of current flow. And it was a really ideal site because the current flowed along a single axis in and out of the bed, where the direction was determined by the direction of tidal flow. So it was always along that same axis. I deployed instruments for 16 days at either end. So I deployed a CFOX sensor at the inner and outer ends of the bed to measure oxygen and pH, as well as temperature and salinity. And then that instrument also had a sensor to measure our photosynthetically active radiation, or PAR, which is our light availability. And then halfway along the transect, I deployed an acoustic Doppler profiler, or ADP, to measure our current velocity, and that also gave us a pressure measurement for depth. So this is the result of those deployments, 16 days of time series data. You see our pH, our oxygen concentration, temperature, light, velocity, and depth. In these plots, the inner end of the transect is shown in blue, the outer end of the transect is shown in red, and the shading indicates daytime or nighttime. So the nighttime is that gray shaded color. And if we just focus on the PAR, we can really easily see that that's the case where there's no light available at night. And then during the day, we did have variable light conditions. We also saw variability in our physical environment. So at the beginning and end of our deployment, we had the spring tides, which are characterized by a higher velocity magnitude and a larger depth gradient. And then during the middle of our deployment, we saw a neap tide with lower velocity magnitudes and a smaller change in depth over those tidal cycles. If we focus on pH, oxygen, and temperature, we can see that during those daytime periods, which again are the white shading, we saw our higher magnitude values versus lower magnitudes present at the nighttime. And this is a lot easier to see if we look at a daily composite plot. So now on the right, we're plotting those same parameters, but now the x-axis is hour of the day. So each point on these plots represents an hourly mean for each day of the deployment, and then the lines are the mean through all of those days of the deployment. So you can see there's a lot of day-to-day variability, but in general, we're seeing this trend of elevated pH and oxygen in the early to mid-afternoon, and then the lowest values seen at night, which makes sense for photosynthesizers that are going to be elevating our local pH and oxygen during the daylight hours. In addition to these in situ measurements, I was able to use them to calculate flux over this transect. So I'm going to walk you through my one-dimensional control volume approach to this flux calculation. If we imagine a box that extends from the inner end of our transect to the outer end of the transect, which I've labeled 1 and 2 on the map, as well as 1 and 2 on our box, our goal is to determine the change in oxygen in that box over time, so dc dt. And we're going to track all the things that might be changing that concentration in the box. So first of all, we have our flux in and out of our control volume. In this case, since we had that really similar axis, or simple, axis of flow, we only expect oxygen to be gained or lost through the ends, not through the sides. Then we have our benthic flux, our flux to and from that benthic environment along the transect. So everything inside that box, which I've attempted to illustrate through these squiggly lines, that's seagrass, and everything else. And then finally, we have our atmospheric flux driven by the difference in concentration in the atmosphere relative to the surface water. But the piece that we really care about is our oxygen flux to and from the benthic community, that net productivity metric. So if we rearrange our equation to solve for that benthic component and put up the real math and color code it so that you're familiar with each piece that we've walked through, now you're seeing where I'm measuring data, or like where the data from the deployment is coming in. So first we have our concentrations of oxygen, C1 and C2 at either end, our velocity U across the transect, and then we're using our depth measurements or height of the water column at either end as well as the middle. And then finally, we need our total length of the transect, which in this case was 571 meters. The result of this calculation is daily oxygen flux data or oxygen flux data per hour is our units here in millimoles of oxygen per meter squared per hour, but I've plotted it as a composite plot of daily oxygen flux, again, with the hours of the day on that x-axis. You'll see that this data has been filtered. So I filtered the data to meet the assumptions of that control volume calculation. The point colors here represent each of the 16 days of our deployment. So any gap in those data points is where data has been filtered out. And since most of the filtering addressed tidal changes, and since the semi-diurnal tidal cycle advances each day, we're left with a near complete 24-hour record since we're able to fill in a different portion of those gaps each day. And if we compare this to our light availability in a similar composite plot and look at our mean flux, we see that the most positive fluxes are occurring when that light is available during the day, and then our negative flux rates are happening at night when there's no light availability, which makes sense if that photosynthesis is what's driving that production of oxygen and drawdown of oxygen at night. Then if we integrate over our means, we get a net flux for this ecosystem over those 16 days of negative 83.54 millimoles of oxygen per meter squared per day, which at first struck me as kind of odd, since I think of seagrass beds as being a really autotrophic system that's producing oxygen rather than being a net sink. But of course, this is a mixed environment. It's seagrass and other organisms. So we have our seagrasses, but we also have corals and other heterotrophs, which are respiring during the day and could be drawing down additional oxygen during sunlit hours. And now returning to our time series data, since my goal in this project and then in the data you'll see moving forward is to really understand these drivers of change in variables we care about. So in the case of acidification, we're really interested in knowing how pH is changing, knowing what our delta pH is, and if there's buffering happening. And then that's similar to our oxygen flux that we calculated. We can also get our travel time or a metric of residence time from velocity. And if we look at all these variables in the space of a principal component analysis here, so in this biplot where the colors are representing our delta pH, where positive delta pH and the delta is downstream minus upstream, so it changes based on what the tide's doing. Our positive delta pHs are shown in purple, and our negative delta pH is shown in this red or orange color. And then the size of our points is the magnitude, so irrespective of sign. We can look at how these different variables that we've measured affect that, that delta pH metric. So first, if we look at the sign of pH, we see that it's associated with our changes in light availability and with our oxygen flux data, which makes a lot of sense because we expect pH to be buffered during times when DAC is taken up, which should be highly correlated with our oxygen flux driven by photosynthesis. And then of course, photosynthesis is driven by our light availability. So we really see that sign set by metabolism, which is driven by light. And then if we look at our magnitude, we see that magnitude is additionally set by that physical environment. Typically, the higher magnitude values in delta pH are at times when we have this higher residence time, and then times, if we follow these arrows backwards, lower depth, so lower volume of water, lower velocity, which of course was also used to calculate our travel time. So we see that the physics really set the magnitude of that sign that we observe. And this is what I want you to remember from this first part, is that the benthic metabolism is really important. It drives our deal variability. And in this case, it seems like primary production has a big role, but physics are also extremely important to account for because they're determining the magnitude and timing of those local metabolic signals. So moving forward, I want you to remember those two drivers, our benthic environment and our hydrodynamics, and their effects on local biogeochemistry. And now we're going to scale up from Palau to basin-wide consideration of data, where we're hoping to still stay informed by the local benthos and local hydrodynamics. And this brings me to what I've been working on in my postdoc with NOAA at CIMAS, so just across the street, although I saw it for the first time yesterday. The title of this work is Linking Offshore End Members to Reef Carbonate Chemistry through Statistical Modeling of Synthesized NCRIMP, or National Coral Reef Monitoring Program, Ocean Acidification Data Sets. That's a mouthful. Basically, I'm trying to predict what the biogeochemistry looks like in these coastal environments, given specific information about those sites and an offshore measure, either measured or modeled of that carbonate chemistry of an oceanic estimate or measurement. I'm working with National Coral Reef Monitoring Program data. NCRIMP monitors reefs across the Pacific, as well as the Atlantic, Caribbean, and Gulf of Mexico to assess the state of those reefs. So in this map, you can see the reef condition. This slide also shows some of the methods used to track our benthic composition, as well as carbonate chemistry. In my case, I've been dealing primarily with the water sampling piece. So looking at discrete samples collected for carbonate chemistry analysis. These are collected every 3 years in the Pacific sites, and typically every 2 years in our Atlantic sites, although there are exceptions, which you will see. My postdoc falls under the umbrella of a larger NCRIMP synthesis and modeling project, where the first objective was to synthesize the NCRIMP ocean acidification observations available. That work was well underway before I started, so I've been focusing primarily on these second two objectives, which are first to compare our reef OA observations, so those nearshore chemical data set, to oceanic end members in order to infer the processes that are driving the changes between the two. So driving those biogeochemical processes, and then to use the synthesis products from the first objective paired with estimates of local hydrodynamics at these sites to build statistical models that have skill to better link our projection models of oceanic carbonate systems to reef scale ocean acidification impacts, or our local signals. And the piece that I've been working on for the last 10 to 11 months is really this determination of local hydrodynamics. Where can we find available data, or how can we estimate the data that we need to feed into these models? So I've been working mostly with Pacific data so far. However, I'm just about to start jumping into some of the Florida Keys data set. But in the Pacific, NCRIMP samples 38 different islands that cover a meaningful gradient in carbonate chemistry. So this map, the background color layer is aragonite saturation state from GLODAP data. So the bluer colors are, again, our higher aragonite saturation state. Redder colors are lower saturation. And I show this not for you to pay too much attention to these magnitudes, but just to see the enormous spatial scale of this data, and that it does cover these gradients in the relevant chemistry that we're interested in. Each of the points also has aragonite saturation state data in this map that's determined from the NCRIMP data set. At each of these sites, in terms of carbonate chemistry, discrete samples are directly measured for dissolved inorganic carbon, total alkalinity, temperature, and salinity, and then we derive aragonite saturation state, pH, and PCO2. Again, these are our 38 islands, where now you can see some of the islands. Some of them are very, very small atolls. Across the Pacific, again, the points here represent aragonite saturation state. However, again, I don't want you to get caught up in comparing magnitudes, because you'll see later on it's kind of hard to make comparisons without having more data. But notice that we do sample around these islands and cover a good deal of the space. And then again, that enormous spatial scale is pretty impressive in terms of the Pacific data set, because we're covering a lot of regions. And that comes at the expense of not having a lot of temporal data. Because like I mentioned a few slides back, these sites are sampled once every 3 years, typically at the same time of year. So we don't capture the temporal variability at these sites. We can start to compare, though, chemistry across the basin. So this is aragonite saturation state across the Pacific Basin over the last decade. Our rows over here are different year groupings, and then our columns are the subgroups of islands. So we have American Samoa, the Marianas, main Hawaiian Islands, the Northwest Hawaiian Islands, and then Pacific remote island areas on the far right. We see that there are gradients across the basin. So typically in American Samoa, we're seeing these higher aragonite saturation state values, whereas if we look at the main Hawaiian Islands, there's lower saturation state. And these gradients across space are pretty stable in time. The exception is our remote Pacific islands in the equatorial regions, where La Nina and El Nino are driving variation in upwelling, so delivering different concentrations of carbon dioxide, which then impacts the aragonite saturation state that we see. So we see a lot more equatorial temporal variability. The other thing that I forgot to mention on this slide is that the boxes or the bars represent all of our nearshore samples for each of those islands or atolls. And then if there's a triangle present, that represents an offshore end member sample collected further away from the reef. And that's important for what I'm going to talk about next, which is bringing us back to this figure that you're all now familiar with of our property plot of DIC, or salinity normalized DIC, salinity normalized alkalinity, and aragonite saturation state in the background. And now I'm going to show you how we apply this to some of our data. So in that previous plot, those offshore end members were represented by a triangle. Here we have a triangle here as a demonstration, where if we imagine that's our offshore signal, and this is an onshore signal, something had to happen to change that chemistry for us to get that signal on the reef. In this case, we likely had calcification, drying down alkalinity in DIC to some degree, and then in order to get the point further left along that DIC axis, we would need some photosynthesis to further decrease dissolved inorganic carbon. We can look at real data and interpret it the same way. So here we have data for Kingman Reef in 2015. We see there's some drawdown in DIC, but the pattern in alkalinity is harder to summarize. Like we have an elevation in alkalinity, but we also have a drawdown. So there's not clear calcification or dissolution occurring as a full data set here. In Tutuila, on the other hand, it's mostly a drawdown in alkalinity, indicative of calcification affecting that environment. In Oahu, we have a similar drawdown in alkalinity, but we also have those data points drawn off that axis of calcification, indicative of photosynthesis having a role. And then finally, in Kure, we have the opposite happening, where there's a slight drawdown in alkalinity, but a boost in DIC, indicative of net respiration. So we can do these kind of analyses to our heart's desire and get an idea of some of the processes that are at play in these environments, but it does have limitations, because these are the data from that entire island environment and don't capture any spatial changes at a given island. So in Kingman Reef, for instance, there could be separate areas of the island that are calcifying and dissolution or having dissolution occur. We don't know how to split up those data, and it's hard. Our sample size goes down if we do it by site. And then the other limitation is this doesn't really tell us about the magnitude of the metabolism occurring, because we don't know anything about the rate of these processes without having the hydrodynamics. We don't know if it's photosynthesis just going crazy or a slower rate of photosynthesis over a long period of time. Same for calcification or dissolution. So what I'm doing is I'm trying to account for the influence of local residence time in these environments, and also divide our data to address sub-island scale variability, because we know that the reefs on Oahu are not all behaving exactly the same way. And so we really need to address those site-specific differences. To do that, I've started with a case study of Guam. Guam is located in the Marianas region, and it's surrounded by coral reefs. In this map on the left, the pink indicates our coral reef habitat. So just the habitat we're interested in here. And then on the right here, I've included our samples from the NCRIMP dataset for Guam. We have 50 samples beginning in 2011. Here I've colored them by the difference in aragonite saturation state relative to an offshore end member, where red indicates a decrease relative to offshore, Blue indicates an increase in aragonite saturation relative to offshore, which gives us some information. But again, it's really, really hard to interpret site to site differences without having a physical metric to track rates. And so that's where we go on the search for physical data or hydrodynamics data. For Guam, there is ROMS model data available, or data from the Regional Ocean Modeling System, which is an unparameterized model which can be applied to specific geographies. In this case, it's applied to the Mariana region by Dr. Brian Powell at University of Hawaii at Manoa. Guam has data, ROMS model data starting in April 2015. This data is available at 3-hour intervals and at a 2-kilometer resolution, which is actually pretty good for these Pacific islands. That's a higher resolution than what's available in other areas. And then the parameters that I need to deal with are the U and V velocity components to get a velocity field over which to make these estimations of residence time to get a relevant physical metric for interpreting our biogeochemical signals. So we have our ROMS data starting in 2015, and we have our NCRIMP samples, but our NCRIMP samples start in 2011. We have sampling dates in the spring from 3 different missions in 2011, 2014, and 2017, which are shown on this map. You can see which year each of those samples was collected. And this is kind of problematic because 2 of our sampling periods are before our ROMS model data is available. So to address this, we decided to take a climatological approach to residence time estimation, where I pulled all of the data available during those windows in March to April and April to May from every year of ROMS data that's available, and then did residence time estimations for each of those periods and calculated Julian day residence time with which to compare our carbonate chemistry sample data set. To address the spatial scale issue or break down that entire nearshore region of the island, I defined nearshore sectors, which were informed by our chemistry data. So using our DIC and alkalinity data set, we defined these 6 different sectors outlined in white in the map and then filled in here where you can see them a little better. And then I had to translate those sectors into what I'm calling ROM space. So into that boxy gridded environment where I can then use the velocity field data and the velocity vectors we have to make residence time calculations for each of those 6 sectors. In terms of our methods, the first method that I use to estimate residence time is a phase flow method or a control volume approach. So this is similar to the control volume that I designed in Palau, except a lot more complicated because we're no longer dealing with flow along a single axis where you're only keeping track of 2 fluxes. We have to keep track of all of the fluxes along the borders of all of these sectors, all sorts of different vector directions in order to determine our fluxes to and from the ocean, or like the open ocean, as well as our fluxes between each of those 6 sectors. To further complicate the matter, when we calculate our volume flux in and volume flux out, they're not in steady state. And so this leaves us with 3 different metrics or measurements of our residence time estimation, one based on our residence time in or based on a flux in, one based on flux out, and then a mean residence time. And then the last thing I want to mention on this slide is that typically doing these phase flows or control volume approaches, we would track a concentration. So in Palau, we track oxygen concentrations over that control volume. Here, we don't really have enough of an appropriate metric to make that or to do that kind of tracking with the data available. We tried doing salinity budgeting early on, but it didn't really work because it's really hard to account for all the sources of fresh water into the system. And so without a measured concentration to track, we took a second approach tracking particles. So this is my sector emptying approach where we did Lagrangian particle tracking in each of these sectors. I seeded particles in each of those nearshore sectors and then modeled their trajectories using ocean parcels, which is run in Python. So we seed the particles in each box or in each of those nearshore sectors, and then look at their trajectories and measure the time it took them to leave each of those nearshore regions. And we calculate a median and mean time or like to represent that emptying time. And that gives us another metric for the residence time in that coastal area. And as an example, these are a couple or a very small subset of trajectories shown around Guam, where I didn't include very many because otherwise it turns into what I call a spaghetti mess where you can't see the individual tracks. But this does show that on the windward side of Guam, we have this longshore flow that kind of hugs the coast and keeps the particles in that coastal environment longer. Whereas on the leeward side of the island, here, if you seed particles in that nearshore area, they're swept offshore pretty quickly. So they end up having lower residence times. Now that we have our residence time metric, we can evaluate it. And this was what was an exciting point to reach because we could say, okay, is everything we've done for several months worth it, or do we need to go back to the drawing board? So let me orient you to the slide and then to the axes of the figure I'll show you to compare some models. On this slide, I'll be showing you models for a response vector of delta DIC. So again, that's our change in dissolved inorganic carbon between the nearshore environment and our open ocean. In the models included on this example, our residence time metric of use is the emptying mean time that we just talked about. On this plot, the x-axis will be our model scale penalized for model complexity, so our delta AIC. So it rewards the skill of the model, but then also rewards simplicity. And then on our y-axis, we have linear model skill, which is just represented by our adjusted R-squared value. So a worse model will have a higher magnitude delta AIC and lower R-squared. A good model or better model will have close to 0 delta AIC and a higher adjusted R-squared value. So if we first bring up our models that do not include residence time, first we see that the benthic environment alone is a very poor model. And this is just a tier zero benthic composition, which only accounts for calcifiers and non-calcifying algae. So that's really near our red X. When we include the hour of the day when our chemistry samples were collected, the model skill improves dramatically. And then if we include PAR, we have further improvement when incorporating that light availability. Then if we include our residence time metrics in the model, we see a further improvement of model skill, with the exception of, so if residence time is independent, the skill decreased slightly from that other model that had PAR and our time of day. But when we include residence time as an interaction with our benthic environment, model skill increases, and that's accounting for the fact that the benthic environment and that time in the benthic environment are very linked. So if you're spending more time in a photosynthetic environment, we'll expect a bigger impact on that dissolved inorganic carbon. And then finally, we have what is our simplified model, including that residence time as an interaction variable. And that's after running model simplification, which would only keep the factors that increase model skill. So that's one example for a response variable of delta DIC. What about our other response variables? And then what if we look at a larger comparison of models using all of those residence times that we calculated? And in this plot, the axes are the same as our previous, or in these three plots, the axes are the same as our previous plot with that model skill penalized for model complexity on our x-axis and our adjusted R-square for model skill on our y-axis. And I'm not going to specify what is in each model, just which models include residence time and which do not. So if we first bring in our models that do not include residence time, and then if we add our models that include residence time on top, we're typically seeing an increase in model skill if we account for residence time. And this is including those 5 different estimates that we've done so far. This is really exciting because right now this is the preliminary test of these residence time metrics, and it's based on only 50 samples for carbonate chemistry. So we expect that these results will get much better and that we'll be able to train a better model as we incorporate more of the data in the NCRIMP data set into our calculations and into our modeling. So for right now, I've shown that we have multiple estimates of residence time, which we can apply to areas besides Guam. And for the Guam data, we've seen that if we include residence time, we do increase our model skill. So our next step is to, and what I'm already doing is moving beyond Guam. So we're incorporating some of the other larger Pacific islands in the NCRIMP data set where there is ROMS data available. We started work on Tutuila in American Samoa, and then we'll do some of the larger Hawaiian islands. And then where we'll really get a whole bunch of samples is when we include the Florida Keys data set. We have almost 1,400 samples from the Florida Keys, so that will drastically increase our sample size. And just to emphasize that difference, I bring you back to this aragonite saturation state across the Pacific Basin, highlighting that we have the Guam data only available for those 3 years, and altogether we have 50 samples. I didn't mention before that the 2020 mission to Guam was canceled due to COVID. It's happening this summer. And then for the Pacific altogether, there are 1,788 samples, so about 1,800. And that's across all of these sites. If we look at the Florida Keys data, and these are figures prepared by my lab mate, Ana Palacio, there's a lot more data where we can do a lot or many more kinds of comparisons with the data available. So this is now a aragonite saturation state across the Florida Keys. We have over a decade of data available. The point colors are year, and then it can be separated into different regions. So she separated it into Biscayne Bay, the upper, middle, and lower Keys. And then in addition to that, like latitudinal separation, we have separation in the offshore extent. So data ranging from our inshore to our oceanic samples. Which this is not something we can do for any of our Pacific Island data sets because there's just not enough data there. And then at each of these, in each of these subdivisions, our sample size is bigger than our sample size for all of Guam in all of NCRIMP, with the exception of this oceanic lower Keys data, which is a little under. And then for the Florida Keys alone in the NCRIMP data, the sample size is on the same order of magnitude as the sampling across the entire Pacific Basin. So I showed this just to show my excitement at getting my hands on this data and doing the same kind of analyses with a lot more data availability. So in conclusion, I hope I've demonstrated an instance where local buffering capacity could be at play. So in Palau, that seagrass is likely providing some mitigation against acidic conditions already, and hopefully with additional ocean acidification stress, that will be the case. I hope I have emphasized throughout that in addition to measuring our benthic processes and accounting for metabolism, we really need local hydrodynamics data. Whether that's measured or modeled, it's something we really need to account for to be able to interpret biogeochemical variability. The NCRIMP records provide an incredible opportunity to make these kind of assessments at a larger scale. In the Pacific, we have that huge spatial extent covered. So we have this great ability to look at those basin-wide gradients in carbonate chemistry, whereas in the Florida Keys, we have that incredible temporal resolution, also really good spatial resolution at that regional scale. And then finally, I hope that through accounting for all of these drivers and for modeling based on those drivers, we can start to use the data that we already have and continue to collect to inform local management to address these global stressors at a local scale and help people identify reefs that have higher resilience and address specific vulnerability on a site-to-site basis. These are my awesome collaborators, both from my postdoc work here at CIMAS and then in my Seagrass Biogeochemistry research at Stanford University. I defended almost exactly a year ago. And with that, I'll take any questions. Thank you so much. Sam.
Want to go first?
Yeah, a very good question. From the Palau, super talk, by the way, I mean.
Thank
From
you.
Palau, from the satellite image, it was about as a seagrass-dominated environment as you could expect anyway. I mean, you could see the dark seagrasses filling that lagoon. But it was still sort of respiration-dominated environment.
Yeah.
I mean, if it's respiration-dominated there, where isn't it? I mean, I was really surprised by that.
So there are some caveats. Oh, does that not select on there? Oh, no, there we go.
I was full of seagrass.
Yeah, so there's a ton of seagrass there. The control flux experiment has a lot of assumptions that need to be met. So if I show that drawing.
Yeah, I remember the.
Yeah.
Yes.
So we have to filter data so that these assumptions hold, where we're assuming that the water that passes one end of the transect is then delivered and passes that second sensor, which means we have to filter out all of the low flow times. So that PCA is based on the data that's kept, but it's cutting out what I think are probably the times when we have peak photosynthesis or peak respiration, because the shallow water is just sitting in that environment, but it breaks the assumptions of the control volume.
Yeah.
So we're not, when we look at that filtered data, that one, when we look at that filtered data, we're taking out the highest, like it's underrepresenting the periods of time that I think would be buffering it most or potentially the most problematic. So this number, I think, has a lot of error on it.
Well, I suppose the point remains that even if it's not all the time it's respiration, it's still most of the time.
Right. And I mean, it's photosynthesis. So like, it's a thing that's not often considered, I don't think, in like blue carbon talk. And when we're talking about these primary producer environments, is that yes, during the day, they are buffering chemistry and can provide this mitigation, but that has to outbalance or like counterbalance the influxing carbon at night, because they are going to be respiring back in the absence of sunlight. And so we really, really need these temporal studies to show the balance and not just take one measurement of pH during the day sometime, where you're not capturing that other side of the process. And then the like physiological testing has to happen in like the kind of work Ian does, where we're looking at these environments and seeing how much that variability matters for organisms, because is it the net number that matters if it's a balance, and or is it the magnitudes of those signals that that matter? The metric that we're considering, we need to interpret in like a physiological sense too, which is why I think we need more of these high institute data experiments to have that kind of knowledge of the variability that's seen in these environments. But we can do a lot with the discrete sampling approach too, because this is kind of the two parts of this talk are kind of the opposite or different approaches to the same question, like really diving into one situation or one site and variability there versus snapshots of reefs around the world and seeing what that data does for us. So there's a place for both of those data in answering those questions.
Yeah. Can I add a comment? I mean, that was like way better than my response. But you're talking about there was a lot of seagrass. So seagrass obviously has seasonal cycles, and senescence and health might be incredibly important. Also, there are all sorts of epiphytes that can significantly impact. We've seen calcification in seagrass beds specifically because of those epiphytes, which is really interesting to think about. So there's a whole, seagrass bed, obviously, the seagrass bed is the seagrass. But also for the blue carbon thing, the only thing that matters, in my opinion, for that is varying and rising. So, it's the overall actual. The
net storage piece, which is not what not talking about today.
Raise and eat it out, and all sorts of things.
Yeah, it's really a whole community measurement here. Yeah.
Did you, is there a flux of oxygen in or out of the sediments? Or did you measure that?
We didn't measure the sediments, so we only had the measurements at either end of the bed. So they're representative of, we considered them representative of that control volume over the entire transect. There are a lot of different approaches to like measuring productivity in these environments. So if you do like the like chamber studies, it's easier to quantify the like sediment flux or different pieces. Then you could also really, really nail down what exactly is contributing to that metabolic signal. Whereas this one, we did some transects of like the benthos that was present along the transect, but over time things are changing. But yes, the sediment fluxes are also contributing to there and not accounted for directly.
Florida, because I don't know how familiar you are with the Florida, South Florida environment, but there are all these seagrass dials in Florida Bay over the years, and also outside Florida Bay on the reef, inner reef tract, and a lot of that is, they don't really know. There's some theories, but one is that there's a buildup of organic carbon and then hydrogen sulfide,
Like anoxic.
toxicity, and this kind of stuff. So it seems like just looking at your study that depending on where you do your seagrass study and the amount of blue carbon essentially in the sediment or the amount of carbon there will give you a different sort of impression of the contributions of seagrasses.
Oh, yeah, absolutely. I mean, it's what Ian said. A seagrass bed is a seagrass bed. They're totally different environments. And that's a limitation of this kind of study, is that it tells us a lot about this particular environment and this particular bay and this type of the world, or in this part of the world, over that 2-week period. It's not representative of the rest of the year or year to year or different sites, which has been something that's been kind of cool for me in my postdoc, is I went from a PhD that was really site specific. So doing this for Palau, doing kelp forests in Monterey Bay, to doing these basin-wide comparisons where you have to start thinking about how these environments vary on different scales. But yeah, that is something we have to be cautious about, is interpreting the patterns we see in one environment and applying them to an environment that may be very different in terms of its drivers and features.
I just have a quick question. Yeah, from the modeling perspective, you mentioned like complexity versus scale. How did you measure complexity? Is it the sparsity of your linear model?
Yeah, so complexity really at this point, and it's just the linear model, it's just the number of parameters that were included at this point, which is not very many yet. We did try running it with a lot more detail, and that's where AIC becomes a lot more important, because we were absolutely overfitting the data for 50 samples if we're including like all of the benthic specific data. So that's why we did a pretty simple benthic composition. But as we include a greater sample size, we'll be able to use the full data sets that are at our disposal to add more detail without. Yeah. So it's pretty simple right now. Our goal now was just to assess, like, is this a relevant, like, does this seem feasible for residence time? And does it look like it impacts our local chemistry signal? Because we didn't know if those metrics for residence time were going to be successful or reasonable.
Yeah. So I mean, so you estimate the parameters, and then you have like the error bounds or confidence bounds for those parameters as well.
Yeah, we need to go more into that. We haven't dealt into that detail too much, since this was the first kind of pass of like, should we continue with these metrics and publish them, or should we go back to square one and do something entirely different? And luckily the answer was like, this seems to be an okay thing. So now we're actually gonna rerun Guam after tweaking some of the things with our velocity input data, rerun Guam, and then we'll do like full uncertainty tests and everything then. So this is the preliminary proceed stage. Okay.
Yes, I don't, is there any other questions? Okay, we'll finish around 3. I hear,
I have questions, but
so I heard NOAA's got a new grant called picogram to study individual coral organismal growth mortality. How do these biogeochemical models tie into the program, and specifically the policy components?
Yeah, so I think policy makers are really interested in if we can get to where we can predict the input here and change it to forecast different situations, I think that'll be really applicable to policy. So if we have metrics of coral presence or absence, percent cover, metrics for different growth, all all of the metrics that are going to be included in that picogram data set as inputs and then tweak them to say, okay, this is how the reef is behaving now. The model is trained on this situation. If we change it and increase calcification this much or add primary producers to this environment as well, how does that change the chemistry that that reef is now experiencing? And does that boost its resilience or does it increase its vulnerability? So that's an aim I have for this, is being able to make those predictions and forecasts. And I think having that better knowledge of the benthic, both the benthic composition and the benthic processes at play are critical for making those evaluations, because we need to ingest as much information to train that as we can to then take in the modeled oceanographic or modeled open ocean chemistry that we have and predict, given a given environment and the processes occurring, what's the chemistry that it's going to experience or be exposing other reef components to.
And one follow-up question,
Yeah.
the hydrodynamic variability impacting this. I mean, is it sufficient to have very high resolution bathymetry and couple that with a 3D hydrodynamic model to constrain that, or is that insufficient?
I think bathymetry is a good step. That having the hydrodynamic model gets more at our time scale. So the bathymetry helps us start evaluating, like, okay, it definitely gets us at that volume piece, right? Which is going to be very important. But I think we need to try to put timescales on it. So if you paired bathymetry with the like benthic volume or like composition that you can get, and then also did ROMS modeling for velocity on top of that, I think that would be covering all of the bases that we need. But bathymetry, at this point, we haven't included bathymetry as a separate variable. We're just using it in the estimate of those volume fluxes. So bathymetry actually isn't used at all in the particle tracing approach, and I think it'll help to increase it. But getting at that time metric, I think, is really important, because how long is a given amount of water in X benthic environment where it's being influenced by Y process? But I think so far, having modeling data for that in ROMS models has been useful. So we can fill that gap. Ian.
So do you have any correlation, spatial correlation between actual performance and Guam and predicted power?
Not yet.
I mean, you can't tell if, like, in one area one is doing better than another, and on one side of
For the chemistry?
for the predicted power of the model. Yeah.
Not yet. We haven't changed anything and predicted anything yet.
Yeah.
We think that in terms of residence time, which is what, again, I've been thinking about the most, it seems reasonable given what we know about flow around Guam and then what other people know about reefs on Guam. I haven't been to the reefs on Guam, but it seems to check out, but we haven't assessed it too much on the site-by-site scale yet. And I'd like to make it more site-specific than those 6 sectors, because even there, it's like the whole eastern side of the island.
Have you thought, and I mean, it's probably not as important in Guam, which is a relatively small island, but have you thought about how to incorporate terrigenous inputs and or groundwater, especially in South Florida, we've shown coming out of Florida, Miami, and Biscayne Bay, groundwater, high alkalinity.
Yeah, I haven't included that as an input yet. I will have to for when we do this for the keys, because yeah, it'll influence our alkalinity for sure. And then also I suspect that having that influence of like sedimentation or reduction in light availability will also be a driving factor that we'd have to consider. So I think there are a lot of other sources of data that we can add, especially in areas that are really well sampled, like the Florida Keys.
To Dave's point, I wonder if to do a high resolution bathymetry, adding changes, with essentially for lack of a better word, I'm not sure there is, the fuzziness of the substrate changes probably like the benthic boundary layer and the residence time, the actual amount of time that the benthic community
is in contact, yeah.
it's in contact. I would imagine.
And like that
It's
kind of
wide-
friction on it, right?
Yeah,
Like
fuzzy.
slowing down the water.
Whatever, yeah.
Steve Monismith's group took our data and did a full hydrodynamic model to look at that question about
Yeah.
residents. So that's why I'm curious if that was like.
Yeah, I mean, I should talk about it with Steve. So Stephen is on, like, he's on this project with me right now because he informed a lot of the early thoughts about control. I mean, obviously, he was on my dissertation committee too. I was like, who do I talk to about control volume? Stephen Madison. So I've had some discussions with him, but they were early on when we were coming up with those methods. And I think it's definitely worth returning to conversations with him to be like, okay, how much do these other smaller scale kind of metrics play into what we're trying to predict? The other thing is for the keys, I think we'll be able to do this, to do sectors on a higher resolution basis to like smaller sectors with estimates for each smaller area. So those kind of metrics will become more useful. Whereas now if we're doing everything at the sector level, like 6 nearshore regions around Guam, having a metric for, even for like runoff, might not be more helpful than not having it because it's probably affecting a subset of that sector, and not including it is of course ignoring it for the sites that are affected, but including it is assuming it's affecting sites that may not be affected. So when we get into these areas where we have better data on both spatial and temporal scales, I think we'll be able to get more specific with the predictions. But it is kind of cool that it looks like we can start doing these predictions on areas where the data is more sparse. So taking advantage of these data sets that are more spread out in time and space and what we have there, and then maybe informing what's measured moving forward in those areas to improve our knowledge or models moving forward.
All right, I think we'll wrap up the questions there. Thank you very much again, Heidi.
Thanks.